students. So here it is your Arka sir. So I have come with a fresh new episode on biology. So if you see my previous episode where I uploaded some very crucial topics on biology, so on human blood, okay. So human blood I already uploaded the plasma, the component, the function of component in details, the WBC, RBC and platelets. In the last episode I already discussed the details of platelets, clear. So and also give you some idea about the classification of blood, okay, A, B, AB and O. So we will continue further, and today you will learn some very important topic on human blood. That means what is agglutinin, agglutination, and why the AB is known as the universal recipient as well as the O is known as the universal donor, okay, and what are the RH factor, anti-RH factor, what are the if any positive blood group given blood to any negative blood group, what happened if a woman is pregnant and if her blood group is negative and the child who is there in her uterus is his blood group, his or her blood group is positive, then what are the complications can come, okay. So these all things, and what is Bombay blood group, a rare type of blood group, as well as the Mn factor, okay. So what is that, clear. So this part only we will discuss today, and in my next episode I will go to the lymph or lymphatic system, clear. So now first let us now start what reaction in between and agglutinogen and agglutinin. So the agglutinogen, it is present in RBC, that I discussed in the previous episode, and agglutinin it is present in plasma, okay. So here it is A and B, and it is alpha and beta, clear. So agglutinogen is present in RBC and agglutinin is present in plasma. So what happened actually, if the alpha A agglutinogen is present, okay, in RBC, and the blood is transferred to a person who has alpha agglutinin in plasma, then what will happen, the RBC agglutination will happen. Agglutination will happen. That means the RBC will react with the alpha agglutinin, okay. The agglutinogen A will react with alpha agglutinin, and what will happen, the agglutination will happen, will take place, and RBC will form, the blood clotting will form, clear. So RBC will break and the hemoglobin will come out, so that will form. So what will happen, the complication will arise immediately. So if the blood in RBC have A agglutinogen, then it cannot be transferred to the blood where the alpha agglutinin is present in plasma, clear. Similarly, if B agglutinogen is present in RBC, in B agglutinogen is present in RBC, then it will react with beta agglutinin present in plasma. So and agglutination will occur, will take place, clear. So that we have to take care of. So that means in our RBC the A and B agglutinogen, if it two type of agglutinogen may present, and in plasma alpha and beta agglutinin, okay, any one of these two or both of the two agglutinin may present. So as per the presence of this, we can give the blood or transfer the blood to other bodies or other patients, clear, who require the blood. So now I will discuss in detail. So hope up to this the reaction between the agglutinogen and agglutinin you can understand. So let us now move to the next topic. Now students, here I have given which classification of blood can be transferred to the other body when the agglutination will not occur. Minus sign means no agglutination will occur, and plus sign means agglutination will occur, okay. So agglutination will occur, clear. That means what? The clotting of blood will occur. Clear? Plus sign. So O in plasma. Okay. So it can be here in case of the O, if the alpha beta or alpha beta agglutinin are present, then no problem. Why? Because the O blood group don't have any agglutination. Okay. So or any antigen present in RBC. So that's why it can be given to all the other groups. Clear? Because so no agglutination will take place. Now A clear. So if alpha or alpha beta agglutinin are present, then what will happen? Then this will react. Clear? Beta if beta and B blood group is beta or alpha beta agglutinin are present in plasma, then what it will do? It will react. Okay, because the B agglutination is present over here. Okay, A and AB alpha beta or alpha beta. If any type of agglutinin is present in plasma, then the agglutination will take place. Clear? So that is why it cannot be given to any other blood groups. Clear? So O can be given to all blood group because it in plasma no agglutinin are present. Clear? And next one is what? AB. That's why O is known as universal donor. Okay, so it is known as what? Universal donor. That means it can give blood to any other blood group. Okay, but it cannot take blood from any other blood groups except its own. That means only O can take blood from itself. Clear? So it cannot take blood from any other groups. Now AB can take blood from all other blood group. Okay, so it is known as universal recipient. Clear? So it cannot give to blood except its own group, but it can take blood from all other blood groups. So that is why the A is known as universal recipient. Clear? Now I am writing one chart. I am writing one chart where you will come to know which blood group can given blood to which. Clear? So it is blood group it is can give, can take. Okay, so first O can give what? O, A, B, AB, and can take only from O. Clear? Now A can give AB, okay, and A. Clear? And can take from O and from itself. Clear? B can give AB and B. Clear? And can take from O and B. Clear? O can give to anyone. Clear? That's why O will be common. Now is AB. AB can give only to AB. Clear? And can take O, A, B from O and from itself also. Clear? AB. Clear? So it can take the blood group, blood from all other blood groups, but can give only to AB. Okay, that's why it is known as universal recipient. And O can give to all other blood groups, but can take only from its own. That's why O is known as universal donor. Clear? So students, hope this chart will help you. Clear? O can give to all other blood groups and take from only from its own, and AB can give only to its own and but can take from all other blood groups. Clear? Now A can give AB and A, clear, but can take from O and A. B can give AB and B and can take from O and B. O can be given to any blood group. See, O is common. Clear? It can be given to any other blood groups. Clear? That's why it is known as universal donor. Clear to all of you? Okay. Now let us move to the next topic. Okay students, now here we will now learn how to determine the blood group. If you have a blood test, how to determine the blood group. Okay. So here two components are used, that is known as anti-serum A and second will be anti-serum B. Okay, anti-serum A and anti-serum B, or alpha agglutinin and beta agglutinin. Okay, the serum basically will be generated from plasma. Clear? So that is known as anti-serum A. So here alpha agglutinin will be present, or anti-serum B, beta agglutinin will be present. So 0.7% NaCl liquid solution will be mixed with blood and 5% RBC component or sample will be produced. Okay, so in a test tube, what we will do? We have to do 0.7% NaCl liquid solution and blood we have to mix, and we have to produce how much? 5% RBC solution or sample. Now this is the glass slide. Okay, so one drop we will add over here, one drop we will add to the another glass slide. Clear? And now in one we will add anti-serum A, and another we will add anti-serum B. Clear? And here we are adding, it is adding anti-serum A, it is adding anti-serum okay, A, and here anti-serum B. Now suppose if this is coagulated and this is not coagulated, if this is coagulated and it is not coagulated, then the blood group will be A. Clear? If it is coagulated but this sample after adding anti-serum B is not coagulated, then what? The blood group will be A. Clear? So if it is coagulated after adding anti-serum A, the RBC sample coagulated, but this sample after adding anti-serum B is not coagulated, then the blood group will be A. Clear? Then the blood group will be A. Now, so all of you have clear? Now next, if suppose if it is not coagulated but it is coagulated, then the blood group will be B. Okay, so after adding anti-serum B, it is coagulated, but that means react with the RBC and it is coagulated, but here after adding the same anti-serum A, it is not coagulated, then the type of blood group will be B. Now the next, if both the sample coagulated, that means after adding anti-serum A, it is coagulated, anti-serum B, it is also coagulated, then the blood group will be what? AB. That's why the AB cannot be given to any other blood groups. Clear? So it will react and it will be coagulated. Clear? Because either antiserum A or antiserum B will be present, okay, or agglutinin will be present, so it will react and the blood will be coagulated. So that's why it is cannot be given. Now the next one is what? If both the blood are in liquid form only, that means it is not coagulated. After adding antiserum A, antiserum B, then that will be O. That's why the O blood group can be given to anyone. It will not react with the other groups. Clear? As no agglutination is present over here. Okay. So that's why it will not react with alpha or beta agglutinin. Clear? Alpha or beta agglutinin will not react. So that means what? It is O. Clear? So it will not be coagulated at no agglutinogen is present in O. Clear? That's why alpha agglutinin or beta agglutinin will not react with that. So that blood group is O. So that means, student, if antiserum A will react and it will be coagulated, then the blood group is A. If only antiserum B will react and coagulated, but antiserum A is not reacting, then that is B. If both will react and coagulated, that will be AB. If both will not react and will not be coagulated, then that is O. Clear? So this is how the blood test sample to determine the blood groups can be done in laboratory. Clear? If you have the sample of this antiserum A, antiserum B as well as the initial solution, then you can also do it. Clear? It is very easy to determine the blood groups. So students, hope you have understood. So now let us move to the next topic. So students, now let us move to the next topic, that is Rh factor. One type of Indian monkey is there. Okay. So in the plasma, okay, so in the serum actually we have one special type of agglutinogen. Okay, we have special type of agglutinogen. It is known as Rh factor. Okay, so the name of the monkey is Rhesus. Okay, name of the monkey is Rhesus. Clear? So in that serum the agglutinogen is present. Okay, so this type of agglutinogen, most of the 95% of Sri Lankan and most of the human, okay, this agglutinogen is present in RBC, but no similar type agglutinin is present in plasma. Clear? So we will get one special type of agglutinogen, okay, in the serum of the monkey Rhesus. So that type of special agglutinogen is present in most of the cases, okay, of human in its RBC, okay, or red blood corpuscles, but the similar type of agglutinin is not present in plasma. Now see, this agglutinogen is made of combination of 6 agglutinogen. Okay, this Rh factor is a combination of 6 types of agglutinogen. What are the 6 types of agglutinogen? Capital D, small d, capital C, small c, and capital E, small e. Clear? So these 6 type of agglutinogen. So this is dominant and this is recessive. So the three components components capital D, capital C, capital E is known as Mendelian dominant, and the three component what? Small d, small c, small e, it is known as Mendelian recessive. What is Mendelian dominant? So when I will discuss the heredity chapter, then the Mendel experiment, dominant and recessive characters, allele, chromosomes, homologous chromosome, so these all type of things will be discussed, but only for your understanding I am discussing in very shortly. Actually Mendel did one experiment in monohybrid cross, two type of plant, one is pure tall pea plant and another is pure short pea plant. So he made a cross between these, so it will be meiosis cell division, so the gamete will be formed, it will be parental generation. So what will happen in the first, it is parent generation, it is first parental generation, what will happen? All tall plant. So that means what? The two gene, one pair of opposite characters will be controlled by two gene. Clear? So here this is what? This is tall character and small t is the dwarf character. Clear? Now in the first F1 generation, all tall pea plant. That means what? The tall character is dominant, and which is recessive here? The dwarf character. So it is known as the tall is known as the dominant and the dwarf is known as recessive. So if we do the cross between the F1 and F1, then in that case what? 75% tall will get and 25% dwarf plant or pea plant will get. Clear? So that we will discuss in details. Don't worry about that. In the chapter. So here the same thing. So the dominant character are specially expressed in the small in capital letter, and the recessive characters are basically expressed to the same letter but in small. Clear? So capital D is the dominant and small d is recessive. Capital C is dominant, small c is recessive. Capital E is dominant but small c is recessive. Clear? Now the thing is that any three pairs will be there. In Rh factor, three component, three agglutinogen. Agglutinogen will be there, but no dominant with the means capital D small d cannot be present together. Capital C small c cannot be present together. Capital E small e cannot be present together. That means it can be what? Capital D, capital C, capital E, or capital D small c, capital E, or capital D small e, capital E, small e. This can be there, but capital D small d, small e, that cannot be. So the opposite characters, dominant and recessive characters together cannot be present for a particular agglutinogen. Clear? So this will be present. So basically the same type of recessive and dominant dominant and recessive cannot be present in the Rh factor. Clear? Now the Rh factor in which Rh factor in which main agglutinogen or dominant agglutinogen are present. That means capital C, capital D, capital E. Clear? Here D must be there. So in that case the blood group will be positive, okay, and the others recessive agglutinogen are present, then blood group is negative, okay, present in Rh factor, then the blood group will be negative. That means if the main capital dominant agglutinogen capital C, capital D or capital E that are present in Rh factor, then it will be what? Positive. And if the recessive characters are present, okay, that means the characteristics of the Rh factor will not be expressed. The recessive characters are present, then that will be what? Negative blood groups. Clear to all of you? So there is A positive, A negative. If this is present in A, then A positive. If this is present in A, then A negative. If this is present in B, then B positive. This is present in B, then B negative. Clear? Like AB positive, AB negative, and O positive, O negative. Clear? So based on the agglutinogen present in Rh factor, so it will be positive and it will be negative. So hope students you have understood what is Rh factor and what is the positive blood group and negative blood group. So now let us move to the next topic. So students, here now I will discuss one very interesting things, okay. So that is what? Bombay blood group. Bombay blood group. So basically what we know, that 4 type of blood groups are there: A, B, AB and O. Clear? So in the RBC, the antigen or agglutinogen will be present, okay. Now it will be A or B, clear, mainly, clear. Now the thing is that the A and B agglutinogen will generate from H antigen. From H antigen, both the A and B antigen or agglutinogen will be generated, clear. But a special case was found in 1952 in Bombay. First, Bombay, in one person, in his blood no H antigen is present. So from H antigen, either A or B anti-agglutinogen or antigen will be generated, but in his body no H antigen will be present. That means A antigen will also not be generated, and B antigen will also not be generated. So what will happen? So this is a rare blood group. So this type of blood group is known as Bombay blood group. So it's very rare and only 0.004% people or human have this type of rare blood groups, okay. So that is known as the Bombay blood group. That means no A or no B, clear, antigen or agglutinogens are present, because H antigen is absent over there, clear. So that is known as Bombay blood group. So students, hope this special type of Bombay blood group, what is that, you have understood, clear. So it is known as the Bombay blood group. So now let us move to the next topic. Okay students, now let us move to the next topic. Let us move to the next topic. So what happened if if positive blood is given or transferred to a person whose blood group is negative. Okay, so what will happen? So when the positive blood is given to any negative blood group person, then after 12 days, what will happen? The anti-RH factor will be created to that person blood, that means negative blood groups. Okay, so now if second time the blood may be required in the second time, again positive blood is transferred to that same person, then what? RH factor, the positive blood. In positive blood, RH factor dominant, three components are there. It will react with anti-RH factor and blood will be coagulated. Clear? So the complication will be arised, okay, or will arise. Clear? So if a positive blood is transferred to a negative blood, having a person having a negative blood, then what will happen? And within after 12 days the anti-RH factor will be created. Clear? So if in future, in the second time, again positive blood will be transferred to that particular person, then what will happen? Then the RH factor of the positive blood will react with this anti-RH factor, and what will happen? And the blood will clot. Okay, and the complication will arise, and the person also may die. So this is the actually complication. So we have to take care of the positive and negative blood groups. And again another complication may come for the pregnant women. What is that? For the pregnant women, suppose if mother negative blood group, okay, and the child in her uterus is positive, then for the first, then what will happen? Then in mother blood the anti-RH factor will be created. Okay, mother is negative, child is positive. Clear? So anti-RH factors will be created. Now in case of in the first pregnancy, so the child may not have any problem, but in case of the second pregnancy, what will happen? If the child is also positive, then what will happen? This anti-RH will enter to the child body from the mother, and what will happen? The blood coagulation or breaking of RBC will take place, and hemoglobin will come out, come out. So this disease is known as erythroblastosis fatalis. This disease is known as erythroblastosis fatalis. Clear? So what will happen? For the first pregnancy, the anti-RH factor will be created in the mother's blood. But when in the second pregnancy, the child is suppose again positive and the mother is negative, then the anti-RH factor will enter to the child blood, and what will happen? And it will react with the antigen with the RH factor of the child blood, and what will happen? The blood will be coagulated and RBC will break down and the hemoglobin will come out. So the child may, very probability is there, may die in the mother uterus. Also what will happen? The bile pigment will increase. Okay, so the child will suffer from jaundice in the uterus only. Clear? Before the birth. So child may die inside the uterus or may die after the birth. Clear? So this is known as the erythrocytosis, erythroblastosis fatalis. Clear? So this is the complicacy if the mother have the negative blood group and the child have positive blood groups. Clear? So this complicacy may arise. Clear? In case of the second pregnancy. So hope you have understood this. Clear? So these things I will again explain in details, in more details in the when I will teach reproduction chapter. Clear? So now let us move to the next topic. So students, now we will, so how many type of agglutinogen or antigens are present now? ABO groups and RH factor. Okay, so agglutinogen or antigen that are present in RBC. Clear? Instead of this, what is there? There is M and N, two more types of agglutinogen are there. No similar types of agglutinin are present in plasma or antibody are present in plasma. So here it will create the blood group M, N or Mn. Clear? But these two more type of agglutinogens, existence of these two more type of agglutinogens are determined in RBC. But what is the most important thing over here, that here the similar type of agglutinin is not present in the plasma. That's why when we are transferring the blood, clear? So the importance of M and N agglutinogen or presence of M and N agglutinogen we will not considering. Clear? It cannot be, it may not be considered now. But why it is important? It is important to determine the real biological father of a child. Clear? So M and N, this agglutinogen, okay, is required to determine the real biological father of a child. Clear? So this is what? This is ABO Rh factor and 2 more agglutinogen existence can be found in RBC M and N, but the similar type of agglutinin are not present in plasma. That's why when transmitting the blood from one person to other person, we don't have to consider about these 2 agglutinogen present. Clear? Only you have to consider the ABO as well as the Rh factor. Clear? But it is required when we have to determine the real biological father of a child, then M and N agglutinogen, okay, needs a very important role. Okay. Now the last topic is the function of blood grouping. What is the importance or function of blood grouping? So first, blood grouping help us to determine the transmission of blood safely from one person to other. Clear? So it helps us to determine the transmission of blood, that means from which group to which group the blood can be given. So it helps us to determine like that. Clear? Now M and N agglutinogen helps to determine the real biological father of a child. Okay. So in some cases the blood group help to identify the criminals. Okay. So these are basically the advantages of the blood grouping. Clear? So M and N 2 agglutinogen are also present, which don't have any role while we are transferring the blood. Clear? We can actually neglect this, but it has a very important role if we want to determine who is the real biological father of a child. Clear? If that type of situation arises. And the main function of blood group is it helps us to determine, okay, the blood transmission from one person to other person safely, so that the person can be safe. Clear? And also in some cases the blood group can determine to identify the real criminals. Clear? So these are the function of the blood group. Clear? So hope you can pause my video and you can write it. Clear? So up to this today, so I hope that you have understood the topic. So in O blood group, in RBC, no agglutinogen are present, no agglutinogen or antigen are present. That's why it can be given to anyone, because whatever the agglutinin in plasma is there, it will not react. Clear? So that's why the O is known as universal donor. Clear? Similarly, the AB don't have any antibody or agglutinin in its plasma. That's why it can take whatever the antigen or agglutinogen is present in RBC, it will not react, because in plasma no agglutinin or antigen is or antibody is present. That's why it can take the blood from all other blood groups but cannot give to anyone. Clear? Because it also have the agglutinogen in RBC, which will react with the agglutinin of plasma of other blood groups. Clear? So it cannot be given to other blood groups. Clear? So this is why it is known as universal recipient. Clear? So it can take blood from all other blood groups. So hope you have understood this. So students, so next video I will discuss about the lymphatic system and lymph. and after that we will go to the human heart. So hope you watch this video. So if you have not yet subscribed it, subscribe it right now, and also share it to your friends so that they also can get the benefit from it. Okay, so thank you.